In this video, we will discuss about pathogenesis of muscle-eye brain disease. This is the disease usually found in the children's, and so far till now there is no cure available for this muscle-eye brain disease. One of the reasons is that people have not been able to find out the root cause for this disease. Some of the studies are been done to find out the root cause for this disease, and uh, however, they have not come to a consensus yet. And the symptoms that observed in this kind of disease is that people have weak muscles, and brain is also not functioning normally. So this slide just condense some of the studies that are done on this muscle eye brain disease. So this is the normal, we can say, dystrophin that consists of alpha dystrophin and beta dystrophin, that is considered to be a main problem in muscle eye brain disease. That is, it is not functioning normally in the muscle eye brain disease. So let us first understand the normal functioning of this uh, dystrophin. Dystrophin has two parts. One is alpha dystrophin, another one is beta dystrophin. This beta dystrophin is a transmembrane protein, or transmembrane protein usually found on cell membrane, and this alpha dystrophin is a extracellular proteins that binds extracellular protein that binds to the beta dystrophin. And dystrophin is considered to be as heavily glycosylated, and this region that is shown in the green color is the glycosylated carbohydrate. So this both combination of alpha dystrophin and beta dystrophin helps in the signal transfer uh, from extracellular matrix to intercellular matrix. One of the examples shown is that the laminin can interact with salic acid of this alpha dystrophin, and through its interaction with beta dystrophin, it can provide signals to the anti-cytoskeletal network, as it is shown here in the bottom figure on the left side. So when the laminin interact with the salic acid of dystrophin, then via beta dystrophin it will provide a signal to anti-cytoskeletal network. However, it is been found out that in case of mutated, or we can say in case of person who is affected with muscle eye brain disease, this alpha dystrophin is not functioning properly. And more close investigation, it has been observed that the glycosylation in the alpha dystrophin is not functioning properly in the muscle eye brain disease. And since laminin binds to the salic acid of this alpha dystrophin, and which is not available in case of, yeah, sorry, so since laminin binds to the salic acid of alpha dystrophin, which is not available or not properly uh, glycosylated in case of mutated alpha dystrophin, so it will not be able to interact with alpha dystrophin. So further signaling that is to to provide actin cytoskeletal network signals to actin cytoskeletal network will not be uh, introduced in within the cells, and this might further leads to the problem that is called as MEB, that is muscle eye brain disease, as this will create uh, problems in normal functioning of eye, brain, or muscles. And the gene that is responsible for this alpha dystrophin and beta dystrophin is the same gene that is later on been cleaved during post-translational modification. And so with further investigation, it has been found out that when the this POMGNT1 gene is mutated, then this might leads to non-functional condition of this alpha dystrophin. So now it has been speculated, or people now working on identifying or confirming that whether this POMGT1 is the gene that is responsible for muscle eye brain disease. And in order to do so, people have identified the gene from the patients that are affected with muscle eye brain disease, and they have find out that in 6 of the patient, uh, this POMGT1 disease is uh, mutated at different sites. And so this might substantiate their point that POMGNT1 is the gene that might cause a improper glycosylation in alpha dystrophin, or we can say interference in O-manosylyl glycosylation in alpha dystrophin, that might leads to muscle eye brain disease. If you need to understand this story in uh, detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.